Thank you. Thanks a lot, Bilal, for the introduction, and thank you for inviting me. It's really a pleasure to be here. So what I would like to do today is first to say, my understanding is the audience is pretty broad, that you come from different fields. Please do interrupt and ask questions if anything is unclear or new to you. It's more fun for me, of course, if it's more of a discussion. So what I'd like to do today is tell you about some very recent work and ongoing work, which is part of a collaboration primarily with Byron Yu, who's at Carnegie Mellon, and also with Christian Machens at Champalimaud. The work I'm going to be presenting today was the work of a really talented graduate student, João Semedo, and a student in my lab, Amin Zanfakali. So very broadly, what we as neuroscientists are interested in is understanding how the nervous system takes information from the environment and uses it to guide behavior and action. And a really simple example of that mapping from sensory input to motor output is the knee reflex arc, which is shown for you here. So we all know that if you go to the doctor's office and they bang you on the knee, that it's going to cause a jerking of your foot. And that's due to a really simple pathway. Why can I not? Is this too weak? Oh, OK. So a really simple pathway that goes from the knee to the spinal cord and then back to the muscles that control the knee position, the foot position. So this is a really simple input-output relationship. And critically, it's a very fixed relationship. So every time that you give the same sensory input, you get exactly the same motor output. Okay? So fortunately, most of our behavior is not reflexive like this, but we have some control of how we map different inputs to different outputs. A really nice example of that was a recent study from the Newsom lab done by Valerio Monte. This is a monkey performing a task. The monkey task is shown here at the top. So the monkey stares at a screen. Two targets come on the screen, as well as a cue that tells the monkey the type of task it's supposed to do. The monkey then sees a bunch of dots, which vary both in their color composition, from lots of green dots to lots of red dots, and also vary in their motion. So they're either drifting to the left or the right. And then depending on the cue that the animal receives, it's going to either report the color content of the stimulus or the motion content. So here's the evidence for that. On the left here, this is when the monkey is doing the motion task. When the motion is rightward, the monkey almost always says that the motion is to the right, almost never says it's to the right when the motion is actually moving to the left. And importantly, the color content of the stimulus, whether the motion is being carried by red or green dots, doesn't really influence the monkey's behavior. Then you give a cue to the monkey, just change this little symbol at the bottom of the screen, and then sort of an interleaved trial basis, the animal can switch. So now that its behavior is being governed almost entirely by the color content of the stimulus, with very little influence of the motion coherence. Okay? So the critical thing here is we have a fixed sensory input, and we have a very different behavior being driven by different aspects of the stimulus based on the animal's goals. So we'd really like to know how is it that brains, as opposed to spinal cords, allow us to change our behavior in this way on a moment-to-moment basis. And we think that this is going to be intimately related to this question of how signals are struggling with this pointer. Faint. So how are signals selectively and flexibly routed in the brain? OK, so let me tell you a little bit about the system that the Monte study was done in. And this is also the system that all of the work today is going to be performed in, which is the macaque visual system, so the visual system of the monkey. This is a side view on the left of the monkey brain. The labeled areas are the ones involved in vision. So about 40% of the monkey cortex, thank you, is that one. Try. We'll find out if it's me or the pointer. OK. So about 40% of the visual of the cortex is involved in processing different aspects of visual motion. What's shown on the right here is an engineer's sort of wiring diagram of this system, where each box here is a distinct part of the cortex. The lines indicate the number of connections between areas. And so you can see that processing visual information involves more than a dozen different areas. And we know from decades of work that each of these boxes performs not a unique, but somewhat distinct function. So there are areas that are more involved in processing color information and more areas more involved in processing motion information. So one way that you could get the sort of flexible behavior that you saw in the Monte study is by selectively routing either color or motion information through this system by changing on a moment-to-moment basis how different areas communicate with each other. Okay? And indeed, this is kind of a widespread hypothesis in the field that flexible behaviors involve sort of dynamic reorganization of the way in which different brain regions communicate with each other. Okay? It's not just a hypothesis. We, in fact, in many systems, including the visual system, have some evidence that this is true. That is, that the interactions between areas can change on a moment-by-moment basis based on the task. So this is one more study I'm going to tell you about which illustrates that, and it sets up some of the sort of competing conceptual framework for the work that I'll be showing you. So this is, again, a monkey doing a task, stares at a dot, a bunch of stuff comes on the screen. The monkey sees a color in the center. The color indicates to the animal which one of these three stimuli it should pay attention to. And then at a certain point in time, random point in time, the brightness of this stimulus will change, and the monkey has to report the change of the cued stimulus. So this is an attention task where monkeys can learn that the cue is useful for doing the task well. And so like you, the monkeys will allocate attentional resources, whatever that means, to focus on this stimulus at this location in space. Now in this work from the Desimone lab, while the animal was doing this task, they recorded from two brain regions involved in visual processing, FEF and V4. And they're just two of those boxes I showed you on the preceding slide. And what's plotted here is the coherence, or the statistical relationship as a function of frequency, of spikes recorded in one part of the visual system with the local field potentials recorded in another. And what you can see, comparing the blue to the red, is when the animal is attending to the stimulus which falls in the relevant receptive fields, that the coherence or the relationship between these two signals is stronger, specifically in these frequencies between about 30 and 70 hertz. Okay? So again, on a moment-by-moment basis, depending on the animal, whether the animal is attending to this stimulus or a different one, you can see a change in the functional interaction between these two parts of the brain. So this is a signature that something in the brain is changing when the animal's task goals are altering. Okay. So we have hypothesis that brain interactions and changes in those interactions are important. We have some evidence that these interactions are robust, and we can measure them with electrophysiology. So this is sort of the starting or launching point for what we wanted to do. And in particular, we were kind of troubled by one aspect of a lot of this literature, of which the previous study is only one example. There are really a large number of studies that have done similar experiments. And that is that our goal is to understand how populations of neurons in one area interact with populations of neurons in another area. But the measurements and the analysis usually focuses on recording one neuron in one brain area and the local field potential in another brain area. So the local field potential is a summary of the activity in this area, but it's really unclear how the activity of these different neurons relates to this recorded signal of the LFP. So using this as a way to assess inter-areal interactions, while it's informative to tell you that something has changed, really doesn't give you access to the sorts of signals that you want, which are the population spiking responses that are actually relayed between these different populations. Okay? So that's what we wanted to do. We wanted to switch away from individual neurons and LFPs to understand this inter-area interaction problem at the level of spiking responses of groups of neurons located in two different structures. Okay. So the thing that we're after then is, what are the principles that govern how population spiking responses in one brain area are relayed or affect another brain area? Okay. So this is the sort of the outline of what I'd like to tell you today. I'm going to outline for you the approach that we took, that is the electrophysiology experiments, and some sort of descriptions of that data set. And I'm going to talk to you a little bit about the conceptual framework that we adopted to try and address this question. I'm then going to march you through a set of analyses, four distinct but interrelated analyses that get at this question of inter-areal interactions. And they give rise to sort of a new view of how areas might interact differently from moment to moment. So I'm going to discuss the implications of that and compare it to some alternative schemes, namely the ones that involve changes in the oscillation frequencies like you saw on one of the preceding slides. And then if there's time, I'll tell you about some unpublished work building on this framework, looking at feed-forward and feedback interactions in the cortex. Okay. All right. So again, this is our model system. We're going to be using the macaque visual system. We're going to be recording specifically from the first two stages of that system, areas V1 and V2. So these are the first two stages of the monkey visual processing cascade. These are large areas, so each of these is about 10% of the cortex in the monkey. And they're also very strongly interconnected, which is captured here by this big black line. So what we do to record from V1 and V2 is we anesthetize animals. We implant them with recording devices, which are shown here. In V1, we place what's known as a Utah array. So this is a grid of 100 electrodes, 1 millimeter in length, which we insert into the superficial layers of V1. And then at the same time, we lower a system of tetrodes through V1, through the white matter into V2. And specifically, and I'll show you evidence for this in the next slide, we can place these tetrodes specifically in the middle layers where the axons of the neurons that live here project. So we're recording simultaneously from an output population in one brain region and exactly from the downstream target, one of the downstream targets of that output population in V2. Okay. So the yield that we get is typically on the order of about 100 cells in V1. When I say cells, I mean both individual neurons and multi-unit clusters, and we're going to group those together. And in V2, our population size is on the order of about 30 cells. Okay. And then while we're monitoring this activity, we're going to present drifting sinusoidal gratings. The stimuli actually for this talk are not particularly important. These stimuli are effective at driving neurons in both of these brain regions. And what we're actually going to do is show the same gratings over and over again and look at trial-to-trial variations in the response to those stimuli, which I'll dig into in a few more slides. OK, so there are many possible brain regions that you could do this type of work in. But I think there are a couple of real advantages to asking questions about inter-areal communication in the context of using V1 and V2 to ask those sorts of questions. So the first is, V1 and V2 have both been studied extensively, also at an anatomical level. So we know a great deal about the connections that go from one brain area to the other, the layers that they come from, the density of the connections, the degree to which they diverge and converge. So we can use that existing literature to help us interpret the functional measurements we'll be making with spiking activity. We also know that V2 very strongly depends on V1 for its activity. And so the illustration of that is this very old cooling study by Schiller and Malpelli. So this is a little sagittal slice of monkey cortex. They lower an electrode into V2, which is labeled area 18 to confuse you. They show a visual stimulus, which evokes a response in the neuron they're recording. And they then cool the surface of the cortex, this is where V1 is, to transiently inactivate that part of the brain. When you do that, when you inactivate V1, you find that activity in V2 basically goes away. And we know similar results come from permanent lesion studies, that if you damage this part of the cortex, that is V1, you're going to basically remove visually driven activity in V2. Okay. So this is important because it means that when we're recording activity in V2 and we're monitoring this output population in V1, we're really recording from a group of neurons that's functionally important for the neurons that we're measuring downstream. And, yeah.
V2 is not getting direct input from the thalamus?
It is getting very, very little input from the thalamus. So in the cat, I think as you know, both area 18 and 17 can be driven directly from the thalamus, but in the monkey that's not the case. There are a few axons, but they don't seem sufficient to be able to drive activity in V2. Yeah. OK, and then the third thing that's really important is that both V1 and V2, because they're early visual areas, have very nice retinotopic maps. So that means that we can place our electrode in a way that we can sample from groups of neurons that encode information from the same part of the visual space and therefore are likely to be communicating with each other. So this is actual data. The blue dots here are the center of the spatial receptive fields of different V1 neurons. And the red dots are the corresponding data for V2. And the shading here just gives you a sense of the size of the spatial receptive field, or the extent over which the neurons will respond in space. And you can see we can record from groups of neurons that have receptive fields that are very precisely aligned down to a fraction of a degree. That actually turns out to be quite important for most of the results that I'll be showing you. If you record from groups of neurons whose receptive fields are offset, you won't necessarily see this phenomenology that I'll be telling you about. OK, so that's my sales pitch for V1 and V2. Let me tell you a little bit about sort of the basic aspects of these paired recordings. So Amin Zanvakali, when he started these recordings in my lab, did the following thing. He implanted the array in V1. He lowered the tetrodes into V2. And as soon as he encountered cells in V2, he stopped moving the electrodes, recorded activity for a few hours, and then moved the electrodes a few hundred microns, repeated, and did that over and over again over about a 24-hour period. And what's shown here on the right are the cross-correlation functions that relate V1 activity to V2 activity, looking at all possible pairings of a V1 neuron and a V2 neuron. Okay? So these have been filtered in a particular way so that only the brief timescale events are left. What you'll notice is that the cross-correlation functions at a bunch of depths in V2 are flat. That means there's no statistical relationship between activity in V1 and V2 at those locations. But at a subset of locations, specifically these, you can see that there's a sharp peak, meaning that after a V1 cell, when a V1 cell occurs, there's some change in the probability of seeing a spiking event in V2. Okay? And these sharp peaks are evident, particularly in layer 4, which we know from making lesions during these recordings. They're specifically the layers that are receiving directly the V1 input. So only at specific layers of the cortex, the ones that anatomically we know are connected, do we see a statistical relationship in the activity between these two brain regions. This statistical relationship, so the probability of seeing structure like this in the cross-correlation function, depends a lot on the separation of the receptive fields that I told you about. When the receptive fields are offset by a degree or more, the chance of seeing this is basically a chance. When they're well aligned, the probability is still really small. So Amin recorded about 300,000 pairs of cells and ended up with about 300 pairs of cells that were sort of directly connected. But the probability, although small, drops precipitously once you start looking at neurons that aren't functionally aligned. So these sharp peaks are different from the ones that you would observe if you recorded from two neurons in the same brain region. And that's shown to you here. So this is a plot of where the peak of this cross-correlation function is for pairs recorded either within two neurons recorded within V1, two neurons recorded within V2, or one neuron in V1 and one neuron in V2. So when you record pairs of neurons in the same brain region, what you usually find is they tend to fire synchronously some of their spikes. And that's consistent with both of those neurons receiving some form of common input. Both A and B are driven by some third neuron, and so some of their spikes will occur at the same time. Between V1 and V2, you see a different structure where the peak of the cross-correlation function is offset by 3 or 4 milliseconds, and that's consistent with neurons in structure A, namely V1, providing input, synaptic input to area B, and therefore elevating the probability that those neurons will fire. Okay? So this is a functional evidence for this anatomical projection that we know occurs between these layers that we're recording from groups of neurons where when the presynaptic side, the V1 side fires, there's a better chance of seeing something happen in V2. Yes?
Is there any feedback from V2 to V1? And also in the correlograms themselves that reflect you had peaks on that were occurring before?
Yeah. So a couple things about that. So yes, there are massive feedback connections. So this is a general feature of the brain, as you know, which is every time an axon goes from area 1 to area 2, there's usually an axon coming in the opposite direction, and that's also true between V2 and V1. Those connections do not go specifically between the layers we're sampling here, but we do later on see some signatures potentially of a feedback interaction, which I may get to. Importantly, the feed-forward and feedback directions, although anatomically they're equivalent in terms of number of axons, functionally, in terms of spiking activity, they're not. Meaning, if you lesion V1, you get no activity in V2, like I showed you. If you lesion V2, although there are just as many axons going back to V1, V1 doesn't really care, as far as we can tell. That is, activity in V1 continues as normal. In terms of that structure that you're seeing, so there is sort of a trough and a secondary peak. Some of that is related to the filtering. Some of it is related to the fact that the neurons have an autocorrelation function. So when you detect a peak, there's a reduced probability of a spike both before and after that spike, and that gives rise to the trough. Yeah. OK. All right, so that's kind of our playground, right? We have these paired recordings in V1 and V2. I've tried to convince you that this is a structure, a pair of structures that are kind of richly connected. And now we want to get to the heart of the question, which is, can we gain traction into understanding, not at a pairwise level, but how a whole group of neurons, for example, in V1, drives and interacts with a whole group of neurons in V2. And the approach that we're going to take to this is to approach this through the lens of a dimensionality reduction approach, where we don't want to fit models that incorporate all possible interactions between each one of the individual elements. And that's because those models are hard to fit and they scale very badly. So as we record from more and more neurons, it becomes more and more difficult to estimate all the interactions. Instead, what we want to do is summarize the activity in each structure with some sort of latent representation or reduced representation, and then understand the interactions through the interactions in this reduced space. Okay? That's sort of the philosophical approach. So let me make that a little bit more concrete, and then I'll show you some specific analyses. So imagine that we have a population of 3 neurons, and we measure the activity of those 3 neurons on a bunch of different trials. So each circle here is the firing rate of 3 neurons in V1 on a different trial. So that activity is summarized by 3 numbers on every trial, but we could capture a lot of the structure of this activity by some form of dimensionality reduction, like principal component analysis or factor analysis, which looks for dominant dimensions which capture most of the variance in this cloud of points. So then we could capture the activity on each trial by the projection onto this line, which is this sort of the activity of that population in this reduced space, or how it falls on this vector. So we're very interested in understanding what we call the dominant dimensions of activity in both V1 and V2. How do we best summarize activity separately in V1 and V2? But again, we're interested in the interaction, so we might, in addition to considering the structure of population activity in each area, want to know how does the activity in V1 relate to the firing rate that's occurring in V2. So now I've taken these same points, but I've just colored them by the firing rate of a simultaneously recorded V2 neuron. And you can see, this is cartoon data, that we've organized this in a way that there's a clear relationship here, meaning that you can predict V2 activity as the projection of these points onto some sort of regression vector. And that this regression vector, which allows you to relate these population responses to the V2 firing rate, is not the same direction in the V1 population activity space as this dominant dimension. Okay? So we're interested in both of these things. What are the dominant dimensions of activity, particularly in V1, and how is that related to the dimensions that are useful for predicting activity downstream in V2? Okay? All right. So here's the analysis then. Let me tell you the specifics of what we've done. So what we decided to do, for various reasons, is to focus not on variations in the stimulus, but at trial-to-trial variability in the response to a fixed stimulus. So I told you we showed gradings. We're going to just choose a grading, show it over and over and over again. And each time we show it, we're going to get in V1 a different population response, because responses in cortex are variable. So this shows you actual data. The grayscale here is a measure of the firing rate. Each square is a V1 neuron. And you can see how the population response varies on each trial. Of course, while we're doing this, we have the same thing in V2, where we record, in that case, on the order of about 30 neurons. And so what we'd like to do is leverage the fact that V1 is doing a different thing on each trial, and so is V2, and to try and understand what aspects of this response can be related to fluctuations in V2. So the way we're going to start doing this is with a simple linear regression approach, a form of regularized linear regression, ridge regression. We're going to count spikes in 100 millisecond bins in both of these areas. That gives us a lot of samples of activity, and then we're just going to fit a multivariate regression model, linear model, to relate these to population activity profiles. So we realized very, very early on that interpreting the outcome of this analysis can be tricky. And that what we really wanted to do here was to learn something specific about the inter-areal interaction rather than the within area. And so we decided for all our analyses to make a comparison between models that relate V1 to V2 to models that relate V1 to V1. So we take this 100 neurons that we've recorded. We take out of that 100, 30 neurons, in this case, that are in each case matched to the V2 neuron. So we have a V2 neuron, we know what its firing rate is like. We go to our whole pool of V1 neurons and we pick out a V1 neuron that does the same thing. We go to the next V2 neuron and so on. So we have here what we call 2 target populations, one target population in V1, one in V2. whose individual firing rates and response statistics, as best as we can do this, are matched. Okay? The only difference is one group of neurons lives in a different part of the brain, one group of neurons lives intermingled with the other neurons that are not in the target population. Okay? So we're going to predict both this group of neurons and this group of neurons with exactly the same population of remaining V1 neurons, which we call the source population. Okay? So these are not only the same neurons, but we use exactly the same data. So the input to the regression model is always the same, but we're either predicting a target V1 population or a population in V2. Okay? All right. So how do we do? So that's the name of the game. We're going to do this with a linear regression model, and here's the first result, which is the performance of our regression model. The performance, all performance measures here are on cross-validated, are on held-out data. So there are 2 points to take from this slide. There's one point that I want you to take, and another point that you'll take despite my efforts. So the point I'd like you to take is V1 and V2 performance is really similar. We can predict V2 activity pretty much as well, in fact slightly better than we can predict activity in V1. The other thing that you'll notice is that our predictive performance is not awesome, meaning that we can predict between 10 and 20% of the variability in either one of these target populations using this source population. So we were a little troubled by this because we'd like models to interpret models that are meaningful, meaning they capture something in the data. But we realized quite early that we were actually doing surprisingly well in capturing activity in these target populations. And let me explain to you why. So this is a really, really dumb simulation in which we create a bunch of random variables. They have properties, meaning variability and correlations. They have statistical properties which are really similar to the measured data. We pass those variables through linear weights, which is what linear regression is trying to give us, to create a bunch of downstream responses. And then we take that synthetic data and fit this regularized linear regression. And what's shown on the right is the performance of the model as a function of the population of neurons that we monitor. For, in the different colors, different group sizes in the predictor population. So specifically, imagine that we have 5,000 of these guys. If we have access to all 5,000, which is over here, our performance will be 100% because we'll get exactly the right model. But if instead of having all 5,000, we take only 100 of the neurons, we actually end up with performance values that are really similar to the ones that we see in the data. Okay? So this means that even when you have a linear model generating your data, if you live in a regime where you're subsampling the inputs, which is very likely to be the case in cortex because we have millions and millions of cells, your performance values are going to be limited by the fact that you have only access to a subset of the inputs. Okay? So our data live here. We're recording on the order of 70 to 200 neurons in our source population. And our predictions are roughly in line with the ones that we actually see in the data. Okay? So it seems that this linear model does reasonably well in capturing the fluctuations as well as we might expect it to based on the sort of the recording situation we're in. Okay, so we have a linear model. We have similar predictability in V2 and V1. And now we want to dig into the structure of this model. And so specifically, we were interested in the dimensionality of the mapping of activity between V1 and V2. So here's what we've considered so far, which is just vanilla multivariate linear regression, where we allow, in the different colors here, each V2 neuron, when we're fitting the model, to just choose how are you going to weight the different V1 neurons. Which ones are you going to listen to? Which ones are you not? In principle, those different regression weights shown here can fully span, in this case, this three-dimensional activity space of the three V1 neurons in this population. What that means is that any change in activity in V1 is probably going to change the projection onto one of these vectors and therefore change activity in V2. But there's an alternative possibility, which is that the vectors that relate V1 to V2 activity don't fully span this space, but instead live in a reduced space in V1. So in this case, we've drawn this as all of the regression vectors for each of the V2 cells, that's the different colors, basically fall in a plane in the V1 source population activity. Okay? That is to say that each one of these vectors must be described by a linear combination of two sets of basis vectors which define this space. Now the reason, if this is true, that this would be interesting is, unlike this case, where any changes in the source population are going to have an effect on V2, in this case, there's a way to change activity in the source population, this dotted line, that has no change in its projection onto this plane. And therefore, any fluctuations in this direction will have no effect on the downstream circuit. Okay? We refer to these as private dimensions because they're not transmitted to the downstream circuit because they don't have a projection onto the relevant subspace that relates activity between the two networks. Okay? So we'd like to know to what degree is the mapping between V1 and V2 like this versus like that. And the way we're going to address that is by using this technique, reduced rank regression, which basically does the same type of linear regression model that's shown here, but does so under a rank constraint, where we can say that we want the mapping either to be one-dimensional, two-dimensional, three-dimensional. It's just a knob that we can turn to decide how much freedom to allow the model to capture the fluctuations that are shared between these networks. Okay? All right, so here we're going to do this for V1, V1. This is an example session. This is the full model, so this is just letting each V2 neuron choose its own regression vector. And what's plotted here is then the performance as a function of the number of dimensions we allow in this reduced rank regression model. So as we allow more and more dimensions, of course if we allow full dimensionality, we recover this performance. What you can see is that although we have a large number of cells in both areas, the performance reaches something similar to the full model after only about 5 dimensions or so. In contrast, between V1 and V2, we see something very different, which is the performance of the model very quickly after just having 2 dimensions approaches the performance value of the full model. Okay. I want to emphasize that throughout the talk and here as well, these numbers don't really mean anything. There's no magic about it. There's no claims here that the dimensionality is 2 in an absolute sense. The dimensionality depends on how many neurons you have and how many data points you have. So the critical thing here is not the absolute numbers, but the relative comparison between these two cases, which I've shown you already is a fair comparison. Okay. So this is one session. This is all the data sets that Joao analyzed. On this axis is the number of dimensions we need in the V1 model to approximate the full model. On this axis, the number of dimensions for the V2 model. And you can see, with just, I think, one exception, that all the points basically fall below the diagonal, meaning the V1 models always need more dimensions than we need for the V1, V2 model. Okay. All right, so we have evidence for a low-dimensional mapping between V1 and V2. But this kind of low-dimensional mapping can arise in a trivial way. So imagine that when we were recording in V2, we had a bunch of V2 cells that did exactly the same thing. We just took a neuron and we just copied its responses. It wouldn't be very surprising then that you would be able to predict that group of neurons with a really simple linear regression model that has just one dimension. So in assessing dimensionality of mapping, you really need to consider how complex is the activity in each of the structures that you're using. And so particularly we were worried that maybe we can predict activity in V2 in a low-dimensional mapping because activity in V2 itself is low-dimensional compared to the dimensionality of activity in this target V1 population. Then the result is basically trivial. So what we're going to do to assess that is we're going to do what I introduced before, which is just summarize the dominant dimensions in each one of these populations. We're not predicting or relating activity anymore. We're just going to take each one of these targets, and we're going to summarize it by a set of dominant dimensions. And we're going to ask how many dimensions do we need to summarize activity either in V2 or in this held-out V1 population. And we're going to do that with factor analysis. So here's the answer. Red for V2, blue for V1. The axis here is the number of dimensions that we need to explain activity in each one of those groups of cells. And you can see that they're largely overlapping. And in fact, V2 is slightly higher dimensional than the activity in V1, meaning that the fact that this is a low dimensional mapping can't be explained by the fact that V2 is lower dimensional than V1. V2 activity is actually slightly more complex than V1, yet the way that V1 relates to it is through this very low dimensional mapping. The axis that's missing here, the y-axis, is the one that we discussed before, which is the dimensionality of the mapping. So this is how many dimensions we need in the regression model to explain activity compared to the number of dimensions we need to just summarize activity in each of those two structures. So you'll notice that the blue points basically fall on the diagonal, meaning that if we have a group of 30 V1 neurons that are basically 5-dimensional, then the mapping from the source population to those V1 neurons is also about 5 dimensions. If that activity lives in 2 dimensions, then we need a 2-dimensional mapping. These two are basically very closely related. But for V1, V2, that's not the case. And even when V2 activity is quite high-dimensional, its relationship to the V1 activity is related through this very low-dimensional mapping. Okay. So there's really sort of a fundamental distinction between the within-area comparison and the between-area comparison here. Yeah.
You may have done this, but the V2, did you do a V2, V2?
We haven't done a lot with V2, V2 just because the population size is really limited. So once we start splitting it up, if you're talking about like groups of 10 cells, you basically always recover one dimension and it's hard to tell what's going on.
But you would expect it to maybe live on the diagonal here?
Yeah, that's right. So we don't think that this difference is related to something specific about V2. It's that this is an inter-areal comparison that's within area. We think but don't know, but we now have data in V4, we could check this, that anytime you look at a within area mapping, is going to be as high dimensional as the group of cells that you're trying to predict. That would be my expectation. Yeah.
When you try to predict V1 neurons from V1 activity, do you introduce a log of 2 milliseconds for justifying a noisy uptick?
No, we don't, but we're using 100 millisecond bins. So the 2 millisecond delay, I think, is, the assumption is it's inconsequential. We've done a little bit of time lag analysis, which I might get to at the end. So it's certainly reasonable when you're looking at small time scales, but for these large windows, I think it doesn't play much of a factor. Yeah.
Have you looked at different subgroups of neurons within V1? Is it possible that you only have two-dimensional dimensions carrying this information forward because you've grouped everything in V1 together and there's really subcircuits?
Yeah. Yeah, so we did worry about that and we did look at that. Actually, we're specifically worried that what if in our models, when we're saying this is our average performance, we're predicting two V2 neurons really, really well and the other one's not at all. So then we get a two-dimensional mapping because we're predicting two neurons, even though when we look at all the neurons, it looks like a high-dimensional representation. So the way we dealt with that is to remove the neurons we could predict the best, the first 2 or 3, because we had 2 or 3 dimensional models, refit the model, and we still get a 2 or 3 dimensional mapping. So both the weights on the V1 side and the predictive power on the V2 side is distributed across all the neurons in the population. Of course, they're not all equally contributing or being predicted, but it's a distributed effect, not a local effect, a couple of neurons. Yeah. Okay. All right. So we've talked about the predictive mapping between V1 and V2, reduced rank regression. We've talked about dominant dimensions of the target population, the factor analysis. And next, we'd like to try and understand the relationship between these. Namely, when we see a low dimensional mapping from V1 and V2, is it just that V2 is only listening to the sort of the most important fluctuations in V1? Maybe it's a sensitivity issue. So the only thing we can detect between areas is sort of the contribution of 1 or 2 dimensions of activity that are dominant. Okay. All right, so the way we're going to do that, I think this is the last analysis, then I'll try and give some context for the implications. So the way we're going to ask this question about the relationship between dominant dimensions and predictive dimensions is by comparing reduced rank regression, which we've talked about, with something that Joao termed factor regression or factor analysis regression. So these are really, really similar. They're both linear regression approaches. They both involve dimensionality reduction. The key difference is then reduced rank regression, you're fitting a regression model, and you're choosing the dimensions of that model specifically to maximize predictive performance. In this case, on the right, what we're going to do is forget about prediction. We're just going to look at source V1 activity. We're going to summarize it with factor analysis. We're going to project responses into that subspace, and only then are we going to do the regression problem. So in this case, the regression dimensions are optimized for prediction. In this case, the subspace is optimized for capturing activity in the source population, not necessarily for prediction. Okay? So if V2 or the target V1 is listening to the dominant fluctuations, both of these should work quite well. If they're not, this one should always outperform that one in terms of performance because this is actually choosing its dimensions to maximize performance. Okay, so here's V1, V1. The open symbols are the dominant dimensions. So we characterize the dominant dimensions, then we do the regression analysis. The filled ones are the predictive dimensions. And you can see that they behave really, really similarly. So it doesn't really matter how you do this, whether you choose your subspace for prediction or for summarizing source population activity. But between V1 and V2, you see, again, something that's quite distinct. In this case, with a couple of predictive dimensions, you do reasonably well. But with only a few dominant dimensions, you do clearly much, much worse than with the predictive dimensions. You need to have a whole bunch of dominant dimensions to start to get the same level of performance you see from the predictive dimensions. These are 2 example sessions, and this is just to show you the summary for the data set. For V1, the number of predictive dimensions is basically related to for performance is the same as for dominant dimensions, whereas for V1, V2, you always need more dominant dimensions to get the same performance you get from the predictive dimensions. Okay, so here's a little summary cartoon just to summarize the observations. We have two cases, between area, within area. This ellipse is meant to represent all the different activity profiles we can see in the source V1 activity. So between V1 and V2, what we have is kind of a narrow mapping, little small pipeline, where V2 is listening to some subset of V1 activity patterns, which are not the most dominant patterns, the darkest shading in the source V1 activity. In contrast, for within area, what we find is a relatively large mapping from the source to the target, and the target is really mostly interested in the dominant fluctuations in the source population. Okay? So we refer to this low-dimensional mapping, as a communication subspace between this activity and that activity. The activity between brain regions might be relayed through a communication subspace. Okay? So those are the observations, and so now let me try and convince you that this might be a really interesting way to think about regulating inter-areal communication. So here's a real, very simple but real simulation in which we take 3 source neurons. Each one of these neurons projects to 2 brain areas, area A in blue and area B in red. So you see there's connections from each neuron to each of the downstream targets. In the case where there's a full interaction, no communication subspace, any fluctuations in this source population are going to generate activity in both of these targets. And that's because of the same thing I told you before, that these vectors that relate, these weights that relate activity in these 2 structures, basically fully span the source population activity. Okay? So we refer to this as a full or open interaction between these groups of cells. In contrast, if we introduce a communication subspace, so again, all the neurons project to both structures, A and B, but they do so through a low-dimensional mapping. Now, as I told you, sort of foreshadowed before, if you have fluctuations in the source population, those may, in this case, be relevant and generate activity in one part of the brain and have absolutely no effect on the other part of the brain, although the axons of these cells project there. And that's because the fluctuations that are illustrated here are like this in the source population. They have a projection onto one of these subspaces, but not the other. Okay? Now, at the next moment, what we can do, for example, we as individuals, if we wanted to do a different task, we would, if we could change the distribution of activity in the source population, with the same neurons, we could now be driving area region B and not having any impact on area A. Okay? So just by rotating activity in the source population so that it falls either on one subspace or the other, we can dynamically regulate which target area this group of neurons is communicating with. Okay? I have not shown you, we're testing this now, that during behavior we do this. We do know from lots of literature that the source population activity can change very easily. There's lots of mechanisms for doing that. So it's certainly possible, and our work showing there's a communication subspace means that those changes may have very different effects on different target areas depending on the nature of the mapping. Okay. All right, so that's our proposal of how inter-areal interactions might be mediated. But there's a competing scheme that I'd like to contrast our proposal with. So this is us. This is the other guys. This is a really dominant hypothesis in the field, which is known as communication through coherence. It's related to the LFP work that I showed you in the beginning, which showed this elevated coherence in the gamma range. So specifically, what Pascal Fries and colleagues have proposed is imagine you want to communicate with a group of neurons C, and you want the neurons in A to be doing effective communication, but not the neurons in B. So you want dynamically to suddenly listen to this, couple these guys and decouple those guys. So what Pascal proposes is one way to do that, or he says the way, is to introduce gamma oscillations in these structures. Gamma oscillations are a certain oscillation frequency where the neurons tend to fire at a particular phase, as shown here. So you introduce these gamma oscillations, and then you make the neurons here and the neurons there have a phase offset that's appropriate for them to communicate. Meaning that when neurons in area A fire, those action potentials are going to arrive at the terminals in area C when those neurons are predisposed to be likely to fire. So the drive is going to be effective. In contrast, these neurons in group B, their spikes will be ineffective because their spikes will arrive in area C when these neurons are not likely to fire. So by changing the phase offset of oscillations in different parts of the brain, you may be able to dynamically link different groups of neurons. Okay? So that's nice in the sense that it maps onto some of the physiology, like the coherence data that I showed you. But we argue that this has some really negative design principle flaws. 2. One of them is that this looks really nice as a schematic, but imagine now that instead of having inputs A and B, you had inputs A through Z. Now your ability to distinguish inputs depends on tiny shifts of phase of different neurons, groups of neurons. These phases are, in this case, are about 20 milliseconds. So you're trying to stack many inputs and distinguish them by very subtle changes in the timing of their action potentials. So we would argue there would be really strong interference between different channels when you're mediating selective communication through an oscillation. In contrast, if you have high-dimensional source population activity, there are many, many low-dimensional mappings that you can stuff into that high-dimensional source population without activity in one channel leaking into activity in the other. So you get here basically a much higher bandwidth across different groups of cells than you do in this case without interference. The other design flaw to set up a brain in this way is that communication here is mediated through time, meaning you're selectively communicating in one epoch of time, meaning you're not communicating in another epoch in time. So your ability to relay signals between the two is limited to specific epochs, and other epochs you have to basically not communicate. In this case, the communication is being mediated by activity patterns across a whole population rather than time. And so we can switch, we can communicate continuously between areas, or we can switch from one communication to one structure and the other without any sort of blackout periods that you would get in an oscillation-based scheme. Okay? So the jury is still out. I don't think that the gamma people have a compelling story. Ours is an observation in anesthetized animals, so we're working on relating this to behavior. But I think as a community, we might need to start thinking not just about the things that we document, but how different signaling schemes might actually be, what the sort of computational advantages and disadvantages are of different signaling schemes in the brain. Okay. All right. Do I have 5 more minutes? Or? Okay. So that's the communication subspace work. And what we're doing now is looking a little bit at the dynamics of the interactions between V1 and V2 during different time periods after stimulus onset. So this is unpublished work. And what we're going to do here is very, very similar to what we've done so far. So far, we've considered these large bins of activity in V1 and V2. We marched them along, and we fit models that relate activity in the two networks. Now we'd like to get a little bit at this delay question, where we start to shift the time epoch in the two areas. So if we take V1 activity in this time epoch, what's our ability to predict activity in the future, and what does that tell us about subspaces? Of course, we can predict in the future, or we can also predict in the past. Okay? All right, so the way we're going to do this, very similar to the approaches so far, conceptually and not that different technically. We're going to use something called canonical correlation analysis. Very, very similar in nature. Unlike reduced rank regression, it's not sort of unidirectional, where one set is the independent variables and the other is the thing you're predicting. In this case, what we're going to do is look for dimensions in V1 and V2, specifically in this case, one dimension, along which when we project the activity in V1 and V2, the activity is maximally correlated. Okay? So we're treating V1 and V2 symmetrically. We're going to choose in each area one dimension, such that when we project onto that dimension, the activity in the two areas is related as strongly as possible. Okay. So this is what that looks like as a function of time after stimulus onset and as a function of delay between the two networks. So values above 0 means that V2 is leading V1. Negative values means V1 leads V2. So you can see something quite stark happens here about 1,300 milliseconds into the trial, and that's the time at which the stimulus turns off. So when we turn off a stimulus, we see a massive increase in the correlation of activity between the two areas. When the stimulus is on the screen, there's a reduction in correlation. The more interesting thing for us, which is hard to tell on this thing where things are scaled the same way, is the time lag of interactions between the areas. So what I'm going to do is just basically take slices through this map, looking at what the interactions are like just after we turn on the stimulus, after some period of time, and what they're like during spontaneous activity. Okay. And those time slices are shown here. So right after we turn on the stimulus, what we see is something that I showed you in the very beginning. We see kind of this sharp peak, which is offset from 0 by a few milliseconds. That means that V1 activity is predictive of what's going to happen in V2 a couple of milliseconds later. As the stimulus persists, that sharp peak becomes weaker and weaker, and things become kind of broad and more symmetric. When we turn off the stimulus, we see this marked asymmetry where actually V2 is more predictive of V1 than V1 of V2, meaning that if we take an epoch of time, V2 is going to predict what V1 does in the future, whereas if we did it in the opposite, V1 would be predicting what V2 did in the past. So this looks very much like activity that might be flowing from V2 to V1, where V2 leads V1, whereas during the sensory-driven response, V1 is driving V2. Okay. So we see a change sort of in the temporal interaction or correlation profile between the areas. And the final thing that we wanted to do is to ask whether this change that we see in the time lag of activity might be associated with communication through different subspaces. So if we choose a subspace, a canonical pair to relate V1 and V2, when they're working in the feed-forward direction, are those the same that work in the opposite direction? So what Joao did is this same thing of canonical correlation analysis. Take an epoch in time, determine what your vectors are, what your two canonical pairs are in the two areas, fix those, and then take those vectors through the rest of the time and see how well you can predict activity or relate activity in those other time epochs. Okay? And he did this in this giant way, which looks at interactions across all time periods. So this is the last data slide. This is the time used for fitting the model, and this is the time used for assessing the correlation. And what you see is lots and lots of yellow when you're using epochs and testing in the same epoch. So during spontaneous activity, we have these feedback interactions. Any canonical pair we pick works reasonably well across other spontaneous periods. But those pairs work really, really badly when you go to the evoked case when activity is going in the forward direction. Similarly, if you take the evoked ones, where activity in V1 is predicting V2, those dimensions are very poor at predicting the interactions during the spontaneous period. Okay? So there are a lot of statistical issues here in terms of doing this correctly. We think we've done it correctly. Joao worked on this for about 4 months. There are very big differences in the firing rates in these epochs. There are lots of sort of trivial ways you might get this result. And we're quite confident at this point that this represents really a distinct way that areas are interacting in the feedback regime versus the feed-forward regime, mainly that they occur through different subspaces. Okay. All right. So in summary, what I'm trying to argue for is that when you study interactions between brain areas, you should use population spiking responses, because those are the signals that are actually relayed between different brain regions, rather than single neurons in LFP. In this case, we use trial-to-trial response fluctuations, and we showed that in V2 they can be predicted by fluctuations in V1. The predictions involve a low-dimensional subspace of V1 activity, which we term the communication subspace. And this communication subspace is not just V2 listening to the dominant fluctuations, because the subspace is distinct from the primary fluctuations within V1. And finally, doing this in sort of a dynamic way, we see that population spiking responses show epochs or periods of feed-forward and feedback interactions that seem to occur through distinct subspaces of activity. Okay? So with that, I'd like to just remind you in red of the people who did this work, and also to thank funding sources. Thank you.
So Pascal's explanation or mechanism, that's a mechanism that really focuses on timing between things. And so there's a direct sort of way of reading things out. And what you've described, though, how is the switching, do you have a conceptual way of thinking of what is the mechanism for actually seeing different or switching between these manifolds?
Yeah, so I agree with the second part. The first part, I don't think it's actually trivial to read out things in time.
Well, it's not trivial.
Yeah.
Agree, but I mean,
But regardless, you could ask the question, what could change source population activity? In the way that we're saying here, different source population activity, in different subspaces causes different.
yeah.
Mechanistically, I don't think we know very well as a field what might drive that. But we do, I think, have really good evidence that that occurs. So across different stimulus conditions, for example, of course that occurs because different neurons have different selectivity, and so the population response profile you're going to get in different situations for different stimulus inputs is going to change. The trial-to-trial variability, which is the focus here, we also know is really malleable. So we've shown that for different stimuli, you can get different dimensions of activity. Other people have shown that with learning or attention, that the structure of population activity changes as those processes unfold. So I think we know as a field that it occurs. We don't really know what the mechanisms are. And we don't yet know as a field whether those changes are really kind of in a goal-directed way to improve communication or whether they're a byproduct of some other process.
Or even at the biological, are you thinking that this is something that has to be implemented at both levels simultaneously, or only V2 is selecting?
Yeah, so I think all these schemes, the gamma scheme a little less, but I think in some instantiations, all of them require what we call an instructor signal. So these are all ways to implement selective communication, but in a way they all require some type of instructor. There has to be someone sitting outside deciding, okay, we want this group of neurons to communicate with that group of neurons. We're going to go in and induce the right phase offset in the gamma scheme, or we're going to change source population activity in the right way to implement our scheme. And what that instructor signal is very unclear. And even, of course, I think most of us think there is no instructor signal. And this has to instead be generated somehow from, for example, the dynamics of these networks. That with some kind of context switching cue, that when you want to use color information versus motion, there's something that goes in and changes the activity within these networks that accomplishes this routing without someone actually looking down on these brain regions and deciding how to wire them up from moment to moment. But all that is super, super unclear in the field in general at this point. Yeah.
Oh, yeah. Sorry. I really liked the talk. It was fantastic. Have you looked at, or do you expect, like one neuron in V2 sometimes listens to a neuron in V1 and sometimes doesn't? Is that sort of like the fundamental of what would go on? Have you looked at that? You mentioned these monosynaptic connection analyses. Have you investigated that?
Yeah, so at a monosynaptic level, I don't know that you would see this. So we have, so what we know is that there are certain population activity patterns within V1 that are effective at driving V2 and some that aren't, and that can be explained through the communication subspace. We do not have the situation in the cartoon, meaning two different target populations and look at switching, and we don't have this for behavior yet. So what we have is, I would call a principle, which is an observation of the nature of the model that relates activity in two parts of the brain, but not yet the evidence that this dynamic routing occurs with behavior or that you can send the signals to one group of neurons versus the other. Right? So all of that is ongoing and future work. Yeah. So I'd say at this point it's an idea and a proposal. Yeah.
I have a question about the feed-forward, feedback part at the end. You may have said this, but is this visual stimulus in this case drifting grading as well?
Yeah.
So I started thinking and worrying about periodicity of the stimulus and sort of what caused the chicken or egg problems. And I think your transients get at that. Early evoke seems to suggest this directional thing. But I'm just wondering, I started then thinking more about natural scenes and things that might be not predictable.
Yep.
So if there is some sort of predictive coding nature to this interaction,
Yeah.
Is there something deeper to say here?
Yeah, that's exactly what we're trying to do with this now. So what I haven't shown you is we have some evidence for gradings that during the evoked period, the degree to which feed forward or feedback is related to predictability. So if you show the same grading over and over, each time you show the grading, the interaction looks less feed forward and more feedback. When you change the grading, you switch to a feed forward regime. So now we're asking, and we've done the measurements, but not yet the analysis, if you take movies, which are quite predictable, because, for example, it's a real movie where the frames have the right temporal sequence, and you compare interactions for that with a jumbled movie, do you see changes in the feed-forward versus feedback interactions based on how predictable the next frame is based on what you've previously seen? So we do have weak evidence of this repeating grading thing for something that might be a little bit predictive coding-like. And we are trying to relate this also to natural movie statistics as well. So that is exactly what we're trying to do. Yeah.
At least for the first part, you were, I guess the first half, you were remarking about the fact that it took more dimensions in V1 to explain its own activity. Given that you're trying to predict the deviations about a mean, and presumably V1 activity is much more strongly stimulus-driven, would you expect that then those residuals to be more white in V1 just because it is so dominated by the stimulus to begin with, and therefore be more higher dimensional?
Yeah, I don't know. So two things, I guess. It's a good question. So, one is the question, to what degree is the trial-to-trial variability related to the stimulus that you're presenting? It seems to be weakly related to that. So if you show a more complicated stimulus, the trial-to-trial variability is higher dimensional than for a simpler stimulus. Then the second question is there a difference in the dimensionality of the trial-to-trial variability in the two areas where V1 is white and V2 is in a reduced space? So that was exactly the motivation and goal for the factor analysis applied to the two populations, which is, let's see, is it the case that V1 activity is whiter and V2 activity lives in a subspace? And that is not the case. So if anything, the V2 activity trial-to-trial fluctuations are higher dimensional than the V1. It's not a big difference, but it's in that direction. So I think the mapping doesn't fall out from the dimensionality of the target, but in principle it could. Yeah.
Okay, let's thank Adam again.
Thank you.